So this is the second part for the coral identification. And I think these are easier to identify. These have more distinct shapes in the overall colony, so they're a lot easier to identify. Again, just remember that with this presentation, it's all pictures, so maybe sometimes you don't really see as many clues as you would if you had the colony right in front of you. So if you have any questions or anything, let me know, but also remember to look in the flashcards and all of that later on, and you'll see that it kind of cements those distinctions that we do for each coral. But just as a recap, what we are looking for underwater. So think of yourself as detectives trying to figure out what this coral is. So you look for clues. Your first clue is going to be colony shape. So how big it is. Is it really big? Is it small, like the overall colony? Then the colony size. That's the size. So shape also means like what. Is it encrusting? Is it making a mound, columns, plates, branching, etc., etc.? Surface is created by the polyps. So is it bumpy or smooth? What does it look like? After that, if you still can't identify, you start looking for smaller things. So you might have to move a lot closer to the colony to see what their polyp size is. So each individual polyp, is it small? Is it big? Does it have the tentacles out or in? That's also a really good clue. Then the polyp shape itself. So where the mouth is and around that, is it round, elliptical, or what kind? After that, remember that polyp color can variate a lot, so it's just going to be a general clue because there are certain corals that only accept a very specific type of algae and it tends to be around the same color. But because the color is given by the algae that it accepts, there are other corals that can have a lot of different colors, so it's going to be a lot harder to tell. So color is a good one for certain corals. As you saw in the lecture before, there are certain ones where we do use color as an identification mark, but it's not an exact science. And then the last one is the septal shape. So just as a little reminder, septa is what we call the little wall where the mouth of the polyp is. So that is the septa, and we'll actually be looking at two corals today that have a very distinct septa, and that's what we are going to use to identify. But remember, this is just like you can always take pictures if you're not sure, and if not, you can just say what the family is, and you can even just say like certain identifying features that it has, and we will help you to try and narrow the identification. So like the corals that we're going to be looking at are just the Caribbean corals, and as of yesterday, all of these corals still remain under the same name, so we are good to go on this. And the way that we use these names, instead of trying to learn the entire scientific name, we are just going to be taking the first letter of the genus and the first three letters of the species name. So for example, here they give us Meandrina jacksoni. So the M for Meandrina, we'll take that, and we'll take JAC for jacksoni. So our coral will not be known as Meandrina jacksoni, but we'll just try to learn MJAC. So this is a lot easier for us to learn. Than like the entire name. If you don't think that you can identify it down to species, then you can only write the first 4 letters of the genus, and this will still give us a lot of information and help us a lot, because we will know around what coral it'll be affecting or what species. So yeah, don't worry about it too much. Just do whatever you can. So the first ones we'll be looking at are some types of meandroid corals. And these corals will have interconnected polyp mouths. So that means that each little polyp mouth doesn't have its own little corallite. So it doesn't have its own little cup where it is, but they all share one big valley. So if you see in this one, you will see where those mouths are, and you will see that there are several that share that valley. Now in this, it's really important, and especially in the coral that they are showing us right now, to look at the septa. So if you remember, the septa is the wall in which the mouth is, so the inner wall of that. So for this mouth right here, this is its septum. Yeah. So what we are looking at is Colpophyllia natans, or CNAT, and it'll be the first coral that we are looking at. I think this previous picture kind of demonstrates one of its identifying features better, and it is the fact that it has this groove right along that septa. So it has this very distinct groove, kind of giving it this this look kind of like a zipper. So it looks like there's a zipper running throughout all of these walls. And that's going to be one of its identifying features. Another thing is that for the most part, these corals will have quite a different color scheme between the walls and the little valleys where the mouths are. So if you see in this picture, you'll see that bright lime green in where the mouths are, and then you'll see this brown along the walls. So that's another thing that it is very distinct in this coral. Another thing is that when the mouth of where the valley of the mouth meets the wall, it'll have a degree angle change in between that. So even if it's all around the same color, it will always have that degree angle change. Another thing is a very complicated word called paliform lobes. So paliform lobes, it's just a fancy way of saying that these grooves along the septum are going to continue down onto where the mouth is. So if you see, you'll have a lot of these grooves continuing down from where that septa is all the way down along to the mouth. Okay, so those are its identifying features. But again, the number one thing that you want to look for is the big groove at the top of the wall, and that gives it the zipper look. So that's your number one identifying feature in this coral. As you can see in this one, even though it has almost the same coloration between the wall and the valley, it does still have its paliform lobes, and it has that degree angle change. For the most part, this coral can be in an encrusting mound, but just like the other corals that we've been seeing before, whenever it has low light conditions, such as when it's at depth, it's going to form thick plates. But for the most part, it's just this big mound. The second coral that we'll be looking at is Manicina areolata. So with our MARE or MARE, it'll have one, and this will help you out in the water if you see a coral like this, because it is quite small. So whereas the Colpophyllia natans that we were seeing before, it can reach up to like 4 meters, like the entire colony. This one, the biggest colony that you'll find will be around 20 centimeters. So it's very, very small, to the point where we really don't see this one as much. And a lot of the times, you will find it free living. This means that it will not be attached to any substrate around, so it won't really be on the reef. A good example of this is along next to the sand patch of the wreck. There are quite a few of these colonies just on the sand. And because of its small size and the fact that the groove along the top doesn't create that distinction that our CNAT does, it gives this coral kind of like a scrunchy look. Because it's so small, it looks like a scrunchie for your hair. So that's what I use to remember this coral. And again, your biggest clue is going to be size when you see this coral. Okay, so as you can see in pictures, it can be a little bit more difficult, but still in these two pictures, you're going to see that the one on the left is going to be our CNAT because of that very distinct zipper line that is growing along the walls, okay? Whereas the one on the right has a very distinct kind of like groove at the top that almost like it's so wide that it almost doesn't look like a groove. And it's very small, or at least like we know it's small, but pictures again don't really do that much justice. And it doesn't have that degree change. It has a little bit of the paliform lobes, but they're not as distinct as CNAT. The next coral that we are going to be looking at is our Diploria labyrinthiformis, or DLAB. So this one also has a groove at the top, but this groove is so distinct that it makes it seem as like at the top it's not actually one wall, but instead it looks like it's two walls chasing each other. So, and it has very, very deep valleys. So you are going to be looking at those two lines that kind of chase each other around. And that's going to be how you distinguish this coral. So if you see all of these pictures are the variations of that exact same coral. For the most part, it grows into a rounded mound, which is why this coral is also known as the true brain coral. And it'll always come in creamy colors, but sometimes the valleys come in very fluorescent color, but the tops of the walls will be in, again, cream colors. So yellow and light browns. But again, just look for those two walls chasing each other. If you see in this close-up, you can see the two, like that it's only one wall, but that groove is so distinct that it's going to make it seem like it's two. So again, you're going to recognize this coral and differentiate it from MARE before because it's so much bigger. The colony itself is going to be a lot bigger. But if you are thinking to yourself, okay, but what if it could be a very small DLAB? In these surveys that we're doing, we're, for the most part, excluding like the very small corals because we're interested in all of the bigger colonies. So don't worry about it too much. But again, the distinction in the groove is going to be your biggest clue. Okay, so in this one, we have our DLAB on the right and we have our MARE on the left. I think the pictures are trying to like make it very obvious, like look at this one is so big and this one is so small. But still that MARE that they're showing us here, you can tell that it's a bigger colony. And that's about as big as they get it. I've never seen one that big. So don't worry about it too much. Just go by size. Okay. Now in this one, you have your DLAB now on your left, and we have our CNAT on your right. So the clue that we are going here is, even though you can see that this DLAB has a color change between the valley and the wall, because that valley is so deep, it's almost not noticeable, that color change. But it's really noticeable in that CNAT, and if we were able to get closer, you would also see the very distinct groove, whereas in this picture of the DLAB, you can see it does look like two walls chasing each other. Now, this is a little bit of a trick question on this one. So it is telling us to remember the identification lecture from yesterday, and it's giving us our Dichosenia stokesi on your right, and it's giving you the DLAB on the left. So if you see these two on the right, it still has those individual coralites for some of them, whereas our DLAB is a Meandroid coral all around. Okay, so any questions so far? I'm going to look at the chat. We are good. No? Perfect. Okay, so the one that we just saw is Diploria, and it used to have another two sister species, but because of genetic analysis, they have now, these two species, been reclassified in a new genus called Pseudodiploria, which is funny because that means fake brain coral. So the first one is going to be our Pseudodiploria strigosa. And this one is going to be our symmetrical brain coral. So it's going to grow into mounds in high light conditions, and it's going to grow in that plate. In lower light conditions, it can grow to be very big, but it's still only around like 1 or 2 meters, the biggest ones. But this one, you're going to kind of work it out because it has the tiniest groove at the top, but it doesn't have a very distinct groove at the top. I'm trying to find a closer image of this. Can't really find it. Okay, in this one, you will see that there's almost no groove at the top. Just the light kind of makes it seem, but it doesn't have an indentation. And it has a lot narrower valleys. So another thing is that it's not going to have paliform lobes. But this one is very distinct from the other ones. I think once you start looking at more and more of these corals, like the difference is going to become more and more obvious as time goes on. So it doesn't have a distinct groove, and that's going to be your main identifying feature. Okay. So here we have the 4 corals that we just saw. Let's see if Caitlin wants to give us a hand in telling us what these are and what you can use to identify them.
Oh, that's a hard one. Okay. On the top left is the DLAB. And you can tell because it has the 2 walls like parallel to each other. And then below that is the CNAT. And that's the one with the little like zipper on the ridge. And it's a different color in the valley and the walls. And next to it is the MARE. And that one is really small and it looks like a scrunchie. And then it's the PSTR, and that one has narrower valleys and ridges.
Yes, this is all correct. So as you can see, once you put them all kind of next to each other, the differences become a lot more obvious. I think like these ones, especially with the close-ups, they help quite a lot. And again, as you start seeing more and more of these corals, it's going to become a lot easier for you guys. Now we have our second fake brain coral, and this is going to be our Pseudodiploria clivosa or Peakley. And it's going to not have as big walls as the other corals that we saw before. So it's going to have shorter walls, and also in the entire colony itself, it's going to have a lot of lumps. So whereas all of the brain corals, as they are commonly called, or that we saw before, this one is going to be a brain coral but with tumors. So it's going to have quite a lot of little bumps, and it doesn't grow in that nice little round shape that we usually associate with brain corals. It'll have more of these lumps just around, and again, it kind of looks like if someone took a rolling pin to those corals before, because it has shorter walls. Another thing is that this coral likes to grow shallower than the other ones, so usually you see these in kind of more of the reef crest, which is not a place that we usually like dive in, because it's shorter than 5 meters even. Here in Utila, if you've been to Waterkey, just as if you've been snorkeling there, you'll see quite a lot of these in the shallower areas. So they are very distinct. I don't think it would be easy to confuse them with the other ones, and it also has a very distinct coloration, as you can see in all of these pictures. It's very brown, or sometimes kind of yellow, but mostly just it's very dull brown color. Okay, so in this one, it's a little bit harder because they're giving you the colony and you can't just look at the finer details. But we know that our CNAT, which is our right picture, it's going to have that very distinct color change. It's going to have that zipper at the top, and I can see those very distinct in this picture. So I'm going to say that's CNAT. Then on my left side, I'm going to have my PSTR. Even though it's a brown color, I don't see any grooves along the top, and my valleys are quite deep there. And then my middle picture, I see quite a few lumps. So that is going to be my Pseudodiploria clivosa. If I had a more close-up image of these, you'd be able to tell like more of the finer details, which is something that we can do if we're out diving or snorkeling and seeing these corals. Now this one is very, very distinct. I love this coral. It's Meandrina meandrites. Very distinct coral. It kind of looks like if you stacked a whole bunch of Pringles on top of each other. And it usually has yellow colors, so it does look like a Pringle. But MMEA is going to have very wide ridges. It's going to have that groove along the wall, but it's almost non-distinct. And it's going to have very, very deep and narrow valleys to the point where unless you got really nice and close to the coral, you can't even see the polyp mouth. But sometimes you will see the tentacles, as you see in the close-up of the image. You can't see where the mouth is, but you can definitely see all of those tentacles. There's a mouth right there. So if you see, it's very aggressive towards other corals. If you see that left picture, you have two separate colonies of Meandrina meandrites killing that poor little Porites asteroides colony right there. So again, this is a very distinct coral. Okay, there is, when it first starts out, it can have certain similarities to MARE, but it's going to have like such distinct thick septa that I don't think you will confuse this, but again, in most of these surveys and whenever we're out diving, we're not really going to be paying attention to these smaller corals. Okay, so if you see very thick, chunky septa here on my left, that is going to be my Meandrina meandrites, and then on my right, I'm going to have my MARE or Manicina areolata. Then we're going to be looking at our Meandrina jacksoni. And our Meandrina jacksoni is going to be kind of a smaller version of our Meandrina meandrites. And by that, I mean it's going to have smaller walls and it's going to have wider valleys. So it's still going to have that distinct septa that it just kind of looks like a roll of coins, but with this one, it's going to have a distinct valley, like you can actually see the valley, and the valley in this one is always going to be white. So if you see in these pictures, you can see that wide valley. If you were looking at these colonies in real life, you would also be able to tell that those walls are really not that high. But just by going with that distinct septal shape and that wide valley, you will be able to tell that this is our Meandrina jacksoni, or MJAC. So if you see in these two pictures, the one on our left is going to be our Meandrina meandrites, because it has the same color throughout and it doesn't really have as much of a valley that I can see. But in my picture on my right, I'm going to have our my Meandrina jacksoni, because it has those wide valleys and shorter walls. But just by looking at there's so much white in this picture, I know that this is going to be my Meandrina jacksoni. Again, there's nothing wrong with this coral, completely healthy. That's just the way that it grows. Okay. Now on these two pictures, I have my Pseudodiploria clivosa on my right, and I'm going to have my Meandrina jacksoni on the left. So a good clue for this is going to be, where do I see these corals? As jacksoni usually grows deeper than my Pseudodiploria clivosa does, or Peakley. And the fact that Peakley has it almost has no walls in this picture, and it has so many lumps. But if you see on my MJAC, it's going to have that very distinct septal shape. So a lot of different clues that are telling me what each of these corals are. Now, this is, I like to think it looks like a sandcastle, but this is the pillar coral, and it grows in a very morphologically distinct way. It creates pillar, hence its common names. And another thing, again, very distinct coral. I don't think there's any way that you can confuse this coral with any of the ones that we've seen before, because it usually has its polyps extended throughout the entire day. And so if you see in this close-up image, it has all of its polyps really extended out. It has really long tentacles. So if you remember to the first identification lecture, we said that our past has this fuzzy look to it, but this one looks really, really fuzzy. Because our past has really small tentacles, but our pillar coral, or our DCYL, has really long, flowy tentacles. Again, this one's very distinct. It grows in that pillar shape. But what happens if for some strange reason those polyps are retracted, as you can see in the picture here? You're still going to be, even if they are retracted, they're still going to be peeking out. So you'll be able to tell that this is your DCYL, and also its shape. If it grows in those columns, it's going to be DCYL. Okay. Now here I have 3 pictures, and they're trying to be like, look, this one has all of its polyps retracted. But you can still tell that those polyps really want to come out. So our middle picture is going to be our DCYL. It has those columns. And then on my left side, I'm going to have my Meandrina jacksoni, because I see so much white in that picture. And then on my right side, I'm going to have my Meandrina meandrites, very distinct septal shape. So really easy to tell them apart. And again, underwater, it's going to be so much easier, because you will be able to look at the entire colony and not just pictures. Okay, so now we are going to start with the cactus corals. They're all in the genus of Mycetophyllia, or MYCE. With these corals, it can be a little bit hard to identify them. Not from like the rest of the corals, but just from each other. We are going to be looking at 3 different types of Mycetophyllia. But again, don't worry about it. If you have a camera, just take a picture and then send it to us, or just write MYCE if you don't, and that is perfectly fine. So we are going to be looking at our 3 different colors. The first one is going to be Mycetophyllia ferox, or MFER. It's going to be a cactus that grows in thick crusts, and it has kind of like a wall all around the coral, and all of these walls grow inwards. So they grow from the outside inwards. Another thing is that you're going to have a very distinct septal margin around the polyp mouth. So wherever those polyp mouths are, they're going to have little bumps all around it. So not just on the walls, but actually on the polyp mouth itself. And sometimes it'll have a different color for the polyp mouth. It's going to be pink. So in Mycetophyllia ferox, it's going to have those pink raised mouths, as you can see in that bottom left picture. And if you go really nice and close to it, you'll be able to tell that it is raised around where the polyp mouth is. Also, the polyp mouth kind of angles outwards towards the edges of the colony themselves. It grows in, as you can see, quite a few different colors. And it can grow around 60 centimeters, so it can be a little bit big. Now, the second Mycetophyllia we're going to see is aliciae, or Mali. Same as MFER before, it's going to have like raised margins around the mouth, but it's not going to have them bumpy like in ferox. And whenever this changes colors, it's going to have a white mouth, whereas ferox is going to have a pink or maybe orange mouth. This one is going to have a white one. Also, the color variation in this one is not as it doesn't come in as many colors as ferox does. It's usually brown, green, or yellow. This one is not going to have as many ridges as in walls as ferox is going to have, and whenever it does have those ridges, it's going to have a lot of kind of like broken ridges, so it breaks down quite a lot, as you can see in these pictures. Again, all of these are the same coral. You can see that it grows ridges, same as with ferox, so it grows them from the outside inwards, but those ridges kind of break a lot and they are not going to intersect. So here we have that very distinct white mouth, so I can tell where all of the polyp mouths are because they are white. They're going to be raised, but they won't be bumpy like ferox is going to be. And this one won't have as many colors as ferox is going to have. here it says that the ridges rarely intersect, so that's what we were saying before. And they break quite a lot, whereas ferox tends to have longer ridges that intersect a lot, and they're not going to break as easily as in aliciae. Okay, so in this one, they're giving me two big clues. So I have on my left side, I have my pink polyp mouths, and on my right side, I'm going to have my white polyp mouths. But even without that, I can see that on my left side, I have those ridges that intersect quite a lot, and I have a lot of walls and ridges just all around the coral. Whereas on my right side, I barely have any ridges, and the ones that I do have are all broken up. So that means that on my left side, I have MFER, and on my right side, I have MALI. I'm gonna see. Yep, no questions so far. We're doing good, guys. And my last one, it's going to be Mycetophyllia Lamarckiana, or emblem. And this one, unlike the other two that we saw before, it's going to kind of want to grow into a mound, so it'll have the edges kind of angled downwards. So that's a good way to tell. Another thing is that it will not have raised septal margins. So that means around the polyp mouth, it's not going to look any different than the rest of my valley. This one also tends to have really thick walls as compared to the other ones. They look very chunky. So here you can see on my left side, you're going to see that dome and those very chunky walls, and all around the valley, I'm not going to see any mouths. So that tells me that it's emlan. And this one, which is an inverted cone, so it looks like a sink. And it has, again, the walls are not as chunky as this one, the same coral, exact same coral. But what gives it away that it's this one is the fact that I see no bumps anywhere that give me a clue as to where the mouths of the polyps are going to be. So I know that is my emblem. So with this one, it's going to have those edges angling downwards, or if they are angled upwards, the center of that colony is going to be inward. So it's going to have like that inverted cone shape. It tends to not grow as big, so it only tends to grow around 45 centimeters, so not as big as the other ones that we saw before. And again, it's not going to be very obvious where that mouth of the polyp is going to be. Okay, now there might be some complications. So some of these corals do hybridize, and others just, like if the colony is small or like where it's growing, it's not getting as much light, so it's going to grow in a different shape. So it might look very similar, so don't worry about it, just write MICE. This is something that it happens to me quite a lot. I'll just look at one and I'm like, oh, this is going to take me a while to figure out. I might just take a picture of it and write MICE on my data sheet. Yeah. Now, in this one, Isophilia sinuosa, it's going to grow in a mound shape. So that's the first thing. It's also not going to grow very big. So going to grow around 20 centimeters, which is, again, not that big when we are talking about colonies that measure 50, 70, 2 meters, 70 centimeters, not 70 meters. That would be a lot. So this one only grows to about 20 centimeters, and it grows in a mound. Okay. Now, it's going to have very, very, very fleshy walls. So it has, even though the skeleton itself is not as fleshy as the walls are going to be, so it kind of looks almost mucousy. And the fact that the septa, so where that wall is going to be, it has these little teeth. It's trying to show us here those little teeth that grow. This one grows in mostly greenish color, so it might be a greenish gray, greenish yellowish, greenish brown. It's always going to have kind of like a tint of green, and these very fleshy walls. So that's going to be your two distinctions. So if you can see here, very, very distinct looking coral, I would say, and it has those teeth. If you still don't know how to identify it, you will be looking at the teeth along the wall. Okay, this is another one that also got reclassified, and it's going to be our Meandroid rigida, or irig. Again, like before, it's going to have very distinct septal teeth, so along the wall, it's going to have those teeth. But this one is going to have something very distinct, and that is that valley is going to be very smooth looking and white. Okay, so the valley is where the mouth is, it's always going to be very, very bright white. And that's completely fine. That's how the coral grows. And again, like distinct septal teeth and that white. Usually it doesn't grow in like long ridges. You can always tell like the little cups is going to have maybe a couple of mouths, but it usually tries to separate each mouth by a wall, like as you can see in this picture. So it's not a meandroid coral per se, but it'll have a very distinct place where each individual mouth is. Okay, so in this picture, I'm going to go by my most obvious one at first, that one in the middle. It has that white. So I'm seeing that white. I'm going to say it's my irig. And then, okay, I have two left. The one on my right, it looks very fleshy. It has very like chunky walls, and it's also greenish in color. So I'm going to say that is my Meandroid sinuosa. And then my one on the left, I'm looking at that dome shape. I'm looking at the fact that it has very wide valleys that I can't really tell where the polyp mouth is. So I'm going to say that that is going to be my MLAM, or Mycetophyllia lamarckiana. So again, another thing that we could go here is going to be sizes. And the pictures are kind of showing us like the sizes, like, oh, look, this one is bigger. This one is not as big as the other one. And this one is really small and tiny. So out in the water, we can also use that as a clue as to which one is what coral. There. Don't worry about these two corals that we are looking at next, because they are solitary and usually free-living corals, so they're not attached to the substrate. These are our mushroom corals. So usually it can be a solitary polyp, but sometimes it'll have more than one mouth. That doesn't mean it's two separate polyps, it just has more than a mouth. Our first one is going to be our Scolymia cubensis, and it's going to have teeth visible beneath the tissue, so not on the tissue itself, like all of the ones that we were seeing before, but you can see it under that fleshy. And another thing is that it's going to be, again, free living or alone, so it's going to be one single mouth with no other something around it. So this single polyp can only grow to like 10 centimeters, which is a lot for a single polyp, but because this is not just the single polyp, but also the entire colony, as you would say, like the entire coral that we are looking at. So it's really not that big, and because of that, it usually doesn't figure into the surveys. And then my Scolymia latera, it's going to have a lot of bumps and very warty looking. So here we have our two Scolemias. Our first one on the left is going to be our Scolymia latera, and then the one on the right is going to be our Scolymia cubensis. Again, you can see it's just one single polyp, and they usually don't tend to grow that big. Here, it's trying to give us that sense of size as well. So lacera is going to be a little bit bigger, but still it only grows to about 15 centimeters. And usually we find them a lot smaller than that. So we really don't take these ones into account into the surveys that we are doing right now. That's not to say they're not important. Okay, now we are going to look at our Musa angulosa, or our mang. I always want to say mango for this one. But this one is going to be kind of like a bouquet of corals, because it looks like a lot of that mushroom corals that we just saw are scolemias, but a lot of them. So this one does grow into colonies. So those colonies can grow to be quite big. They will usually come in like a grayish color, but it'll also have quite a few colors thrown in there. So it will often look mottled. And it only has live polyps at the top. So as we saw before with our Orbicella annularis, that it has a dead base, but the colony itself can be dead at the bottom and still alive at the top, and that's completely fine. That's just the way that it grows. I think that's the only picture that we get of this. We don't really get a picture of the full colony, but it'll just look like a bouquet of that mushroom coral that we saw before. And how you are going to be able to tell that it's one colony of this and not just like a bunch of mushroom corals that ended up in the same place is because these ones will share a base and they will all have around the same color. Whereas if you see a lot of different scolemias, they're going to have very distinct color each and every single one of them. Now, the next coral we are looking at is Eosmilia fastigiata, and this one you will be able to tell by size. So our Musa angulosa that we saw before, it's going to be this bouquet, but each flower is going to be big. So every single one of the polyps at the top is going to be around 8 to 15 centimeters, so quite big. but that's just the polyp itself. Like the colony can grow quite big, whereas our Eosmelia fastidiata, or EFAS, each polyp at the top is only going to be around 1 to 3 centimeters. So again, size is a really good one with this. And just visually, it has very distinct septa, it has a dead base, it has that color, and it sometimes has a very distinct purple color. So this coral is quite unique. And once you see it out in the reef, you're going to be able to tell it right away what it is. So in pictures, it might be a little bit hard, but once you're out there on the reef, it's going to get a lot easier. So here we have our 2 bouquets. It's telling us that, again, just by like the size of the pictures, it's trying to tell us that the colony on my left is smaller than the colony on my right. But I can also see that, okay, these ones, like the polyp themselves, are quite big. So that's going to be my MANG. So the one on the right is MANG, and the one on the left is EFAS. Okay. Any questions so far? Oh, yeah, that's it. Okay, yes, any questions so far? Okay, no questions. So now that you guys already have a kind of a head start on what we are looking for, like what corals we are looking for, Caitlin is going to give us another presentation on the disease that we are looking for and how it presents on these corals. And more of like what we are going to be searching for when we are looking for this disease, because at the end of the day, this is what we are trying to do. We're trying to create a plan and be able to have as many divers and as many helpers out there watching out for the corals and be able to hopefully at some point, if the disease hits here, be able to train a lot of people in being able to treat against this disease. If you guys have any questions about the corals, again, you can ask me, Caitlin, or any of the other people on the group, on the Facebook group, and you can also use the drive folder that we sent before, and that is going to be on the Facebook group. So that you have the flashcards, you're going to have all of these presentations, so you can take a look at them later. And it also has a little cheat sheet. So it has a picture of the colony and then a closer look at the polyps themselves that you guys can use. So now Caitlin is going to give us the presentation. Thank you guys for your attention.
Thanks, Andrea. Okay, so as Andrea said, I'm going to do a little bit more detailed presentation about the stony coral tissue loss disease. Now that we have an idea about what corals we're going to be looking at and looking for. Oops. There we go. Okay. All right, so if during the presentation anyone has questions, please go ahead and ask. Okay, so just a reminder about what stony coral tissue loss disease is. Remember that it is relatively new and very aggressive. It affects 20+ species of our important reef-building stony corals, so a lot of the species that you just learned about. The cause is still undetermined, bacterial pathogen suspected. It spreads via direct contact, so coral to coral, ocean currents, and likely through ballast water. It causes rapid tissue loss, leaving behind large areas of dead white skeleton. As you can see in the photo on the right, that's what the skeleton will look like if it's exposed. The skeleton may quickly become colonized by algae, so it might look kind of fuzzy and it might have a coloration to it, but the algae is going to be very different looking than the actual coral tissue. We still have not yet detected this disease in Utila. That's why we need your guys' help. So we have an early warning and we can catch the disease at the beginning of the spread and hopefully intervene. So I'm going to show you just a couple of seconds of video from the disease outbreak in Cozumel, Mexico. So you have an idea of what it can do to a reef and what it can look like when it reaches the reef. So as you can see, a lot of dead corals. Most of the colonies are mostly dead. You can tell that it's a dead coral because it's white and you can see the skeleton. There isn't any living tissue remaining there. Okay, so that's just to give you an idea of what we're up against, what we definitely want to avoid seeing here in Utila. Okay, so a little bit about how we can tell if what we're looking at is stony coral tissue loss disease. So again, it goes in a species-specific order. So highly susceptible species will be affected first, and we're going to go through which species those are. It spreads rapidly, so once present, the disease will spread quickly throughout the reef. So like you saw in the video from Mexico, a lot of the coral colonies are infected. Mortality rate, so the infected corals rapidly lose living tissue, and the highly susceptible colonies may die within weeks to months. So we can see the top photo on the right here. This is from Florida. In this colony, you can see that it started with a smaller lesion, 5th of January. By January 19th, so 2 weeks later, that had grown. And then by February 1st, less than a month later, it had taken over 60% of the coral colony. Prevalent, so many, if not all, colonies of highly susceptible species on a reef will become infected. It can have multiple lesions, which is different than most other coral diseases. So the tissue loss typically occurs in more than one area of the affected colony. So you can see on this large CNAT colony on the bottom here that it has multiple lesions throughout the colony. Okay, so that is pretty distinctive with this disease. The length of the disease outbreak as well, so it lasts longer and is less affected by seasons and conditions. Okay, so what are our highly susceptible species? So these are the species that are affected first. Okay, so these are the ones we're really looking out for. These species have a very rapid progression total mortality. So could be 1 week for small colonies and 1 to 2 months for larger colonies. So that's really quite fast. So our first coral on the top left here is our Meandrina meandrites. Okay, I'm just going to say the codes because I'm going to butcher the scientific names. So our MMEA. So this is one of the first corals to become affected. Our DSO here in the middle, also first to become affected, and then followed quickly by our CNAT here. Okay, our other highly susceptible species, they're quickly following the first three species. They're also affected with rapid progression and total mortality. So our EFAS here, we just saw this one in the coral ID. Our pillar coral, our DCYL, which I'm sure you guys all know. Our PSTR, or symmetrical brain coral, the DLAB, and the knobby brain coral, the PCLI. Okay, so these are all our highly susceptible species. These are the ones we're really, really looking for at this point to detect the first signs of this disease. Now, our intermediately susceptible species. So these species are typically affected around 1 month after the first onset in the highly susceptible species. Typically, there will be slower progression, mortality in smaller colonies occurring after several months, and larger colonies possibly in years. So these are all our star corals, our Orbicella species. So our OFAB, OAN, and OFRA. Okay, so there's our OFAB there. Our MCAV, the SBU, which we don't really see here in Utila. Our SINT, the blushing coral here, and the SCID, which we do get a lot of here. Okay, so these species we're also looking at. Typically, they would become affected after the highly susceptible species, but we are also looking at these species to see if we find any disease signs there. So some species that are rarely or not affected, so our AGRRRA species, so our staghorn and elkhorn corals, those are not affected. So if you do see disease signs on those species, it is going to be typically a different disease. Our finger corals, so Porides species, also rarely or not affected. So here are some examples of how this disease may look on our susceptible species. So this was put together by one of the foremost researchers on this disease in Florida. Since none of us here in Utila have actually seen this disease in person, we have to rely on descriptions and photos from other places. So I can only give you kind of limited information about how it can look. And these are just some examples. This slideshow is in the Drive folder that we've shared, and I believe it's already been posted on our Facebook page. If I haven't posted it yet, I will post it. So on our MMEA, this is how the disease can look. So you can see it can have slightly different appearances, either the defined margin line on the left, where it's very clear between the living and dead coral. In the middle, you can see that you could also have a bleached disease margin line. So you'll have bleached coral tissue in between the living and dead, and then multiple lesions and merging together. And here on our DSTO, again, you could have one single lesion or multiple lesions looking slightly different. On our DCYL, you'll have many lesions on the colony, different margin lines, and the tissue actually just basically falling off of the coral. Our EFAS, so because you have stalked polyps, it looks a little bit different. So you can see that it's affecting different polyps at the same time. And CNAT, our PSTR, and our PCLI. So on the bottom left, you can see that it has different lesions coming from each side. And the DLAB, again, you can see that multiple lesions on the top left photo. And the OFAV, again, you might have this fine margin line or bleached margin lines. And the MCAV. And on the SCID, there are a lot of different ways that the disease can appear. In one reference, it did say that SCID can show signs of disease very early on in the outbreak, which may or may not be the same disease. And as you can see, it can look very, very different. Some other common coral diseases. So as I said yesterday, I am not at all an expert in coral diseases. Luckily, Danny is, and she will be giving us a presentation more detailed in how to recognize different coral diseases so we can have an idea of how to tell those apart on the reef as we're looking for SCTLD. So some common coral diseases we can see here in Utila. White plague, which does affect stony corals, as you can see in the bottom left-hand photo. Dark spots disease, as you can see in the middle, it is pretty much how it sounds. You'll see dark spots on the coral. Yellow blotch, which I don't have a photo of, and I didn't say black band, that's on the top left. So other conditions commonly observed here are paling or bleaching corals. So like the SCID on the bottom right, as Andrea mentioned yesterday, it can turn like a blue or purplish color when it's bleaching. We might see predation by parrotfish, damselfish, fireworms, or snails. So those creatures are actually eating some of the living coral, and in the case of the parrotfish, some of the skeletons. Competition and overgrowth from other organisms on the reef is also common. So corals that are already stressed by these conditions or other environmental factors may be more at risk for developing SCTLD. Here are some really good ID cards that were made for the Florida outbreak. I've posted these on our Facebook page. You can download them. If you want to, you can also print them out. They're meant to be able to be laminated and taken with you on the dive as a quick reference. They also identify the susceptible species and give you examples of how SCTLD looks on those. And then here you can see the other diseases and how they look, and the other conditions and how they might look. So it's very good, handy reference. I recommend downloading that. So a little bit about how we can differentiate SCTLD from white plague, which is probably the most similar condition. So some of the similarities, most of the same species are susceptible. Both diseases can start at the base or margin of the coral and radiate out. And colonies exhibit rapid tissue loss. Okay, so that's how they're similar. Now, how they're different is lesions may appear within the remaining tissue of colonies in SCTLD in the middle of the colony, as opposed to usually starting at the margin or base of the colony as affected with white plague. SCTLD affects coral species in the specific order. So like we just talked about, those highly susceptible species will show signs first, then followed by the intermediate susceptible species. And SCTLD has continued to spread throughout the reef tract for now 6 years in Florida, and the pathogen is presumably transmitted via water currents. White plague outbreaks tend to be more short-lived and have subsided in winter months as temperature cooled. So that's another good indicator that white plague is affected by seasons and conditions. So another common condition we see here in Utila is coral bleaching. So it's not a disease, and you guys probably know a bit about it already. Some similarities are that corals turn white and may appear in spots or entire colonies and affects many species simultaneously. The differences, so SCTLD is caused by pathogens, and bleaching is a stress response due to increase in water temperatures. SCTLD causes rapid tissue loss, whereas bleached coral remains alive. The color loss is caused by lost algae, but at least initially, the coral does remain alive. And if the water temperatures lower again, the corals can recover and take back their algae. So if you see the photo in the middle, it is a partially bleached coral and it's bleaching in spots. But if you look closely, you can see that the coral tissue is still alive. It's just colorless. You can see it looks fleshy, and inside of the valleys, you can still see that the polyped mouth still have color in there. So it may look like a disease, but if you look closely, you can tell that it's bleached. And then in the example on the right, a coral that was affected with SCTLD is actually dead, and the skeleton is exposed, so the white part has no more living tissue. You can see how it really just looks, it doesn't have any fleshiness to it anymore. So again, corals can recover from bleaching, but they're left weakened and more susceptible to other diseases. So treating SCTLD. So the simplest and fastest treatment, which is most effective on small lesions and corals with an early stage of the disease, is directly applying an antibiotic paste onto the diseased tissue. So this is very interesting. So this treatment has been shown to work, as you can see on the diagram on the right. The top corals were treated with a placebo, so that means there was no medication. Obviously, it did not halt the disease. In the bottom photos, those coral colonies were treated with an amoxicillin, which is an antibiotic, mixed into a base and applied at the line of the lesion. So you can see on the first photos there, it was pre-treatment. Then in the second photos, you can see the actual line of paste where they began the treatment. And then on the third photos, after 1 month, and they revisit the corals, that you can see that that treatment actually halted the disease progression and stopped it at that level. So really, really important that we catch this disease early and try to intervene. If we can intervene with this kind of treatment, then we have a better chance of keeping some of our coral colonies alive. So our mitigation strategy for Utila. So early detection via our monitoring program and our response team divers, which are you guys. So everyone keeping an eye out on the reef, taking photos of any suspicious looking corals, getting advice from experts. So if we do get sent any suspicious photos, we are sending those off to experts to try to help identify and see if it is the disease or not. Also developing a response plan with the Bay Islands National Marine Park co-managers. So that is in the works right now, to have a standardized response plan and protocol throughout the Bay Islands Marine Park. We're also looking at doing a training in the treatment protocol, bringing down an expert from Mexico who has treated corals there and getting trained in the techniques. Community outreach and education. So also working on that, working with the dive shops once everything gets reopened, and hopefully educating more people in the community about this disease and how to go about detecting it and treating it. We also are working on securing funding for future monitoring and treatment. Really, really important, unfortunately, because of the effects of our human disease. Right now, we are really short on funding. So if anyone of you guys has any connections to funding source, please do get in touch with us. That's something that is really, really important and is going to make a huge difference for us going forward if we can secure funding for our monitoring and training and getting the treatment supplies here. Okay, so again, what should you do if you see something suspicious? So hopefully you have your camera with you. Really important, if you can, take pictures. So of the infected area, a close-up of the lesion, a picture of the whole colony, and then if you see any other diseased corals in the surrounding area, getting photos of those and trying to get some photos of the surrounding area. Make a note of the exact location, GPS points if possible, depth, water temperature conditions, and navigation aids so that we can find the coral again. I'm sure you guys are good at that. So trying to help us keep track of where exactly the corals are. Really important to remember, do not touch any infected corals because this disease does spread through direct contact. You don't want to get any of those pathogens on yourself, on your equipment. If you suspect SCTLD, do not dive in a separate area of the reef because we can bring that with us in our equipment to another area of the reef and spread it that way, which we definitely don't want to do. We do want to disinfect all of our gear and remove any debris, sand, sediment, seagrass, anything like that. And I've posted the decontamination guidelines on the Facebook page. There are general guidelines and the detailed decontamination protocol. So that's something for us to be aware of and for our dive shops to be aware of. It will intersect with the decontamination protocol for COVID that we will have in place going forward. So it is really important for both humans and corals that we're disinfecting our gear. And really important, report your sighting. So please post any photos and other information on our SCTLD Response Team page. Oh, okay, I forgot to put this in there. The general guidelines for disinfection. So again, just inspecting the gear and removing debris. If we know that we're doing surveys on sites that do have SCLD, you move from the cleanest site first to the dirtiest last. So least incidence of disease to most. Decontaminate your dive gear, of course, at the end of the day, and also between dive sites, between countries if you're traveling, and between sensitive areas. Properly dispose of disinfectant and rinse water into sink, tub, or shower. That is thinking that that water will go into some sort of treatment facility. As we know here on the island, that's not always the case. So that's another consideration that we have to make, is how we're going to dispose of our disinfectant and disinfecting water, and which disinfectants we're going to use. In the detailed guidelines, it does give specific disinfectants and formulas for the, what's the word I'm looking for? When you mix it with water. So don't leave debris on your dive gear. Don't move from disease to a healthy site. Don't forget to disinfect your gear between sites, countries, sensitive areas, and end of the day. And don't dispose of disinfectant and rinse water into the ocean or storm drain. And again, really important stuff that we can all do, no matter where we are, to help our corals be stress-free. So not using chemical sunscreens, covering up when we can, and only using reef-safe sunscreen, using natural mass defog or biodegradable defog, always using your best buoyancy and teaching your divers to do the same. Really important to avoid stirring up sand and sediment and keeping hands, fins, cameras, gauges off the reef. Limiting our chemical impact at home by choosing biodegradable personal care and cleaning products, considering our carbon footprint and trying to limit our carbon emissions, and again, spreading the word about coral reef health and how we can protect coral reefs, and just generally educating our friends and peers about the dangers facing coral reefs and what we can do about it. So thank you guys so much for your help protecting Utila's reefs. We have the best chance of detecting SCTLD early with a lot of people out there on the reef looking. And early detection means early intervention. So don't forget to use our Facebook page, Stony Coral Disease Info Utila. So that should be your go-to reference for updated information on SCTLD. For all actions specific to our group efforts, we're going to post it in the Response Team Facebook group. Again, if you see anything suspicious, post your photos and observations to the group. And of course, if you have any questions, feel free to ask in the group. And together we can keep our reef healthy and happy.